Jai Hind, welcome to Bioaffairs. So, as you can see in the thumbnail, today I'll be discussing the part 2 of virology related matters. So, in the first part, I have discussed the classification, morphology, and some important aspects of viruses. Now, today I'll be talking about the second best or second important, most important topic of this viruses life cycle, that is replication. So, here I'll be talking about not only DNA, also RNA virus and the subdivisions like single stranded, double stranded, RNA, DNA, and the total process of their progeny replication within the cell, either it is a human cell or animal cell or in a bacterial cell. So if you follow this class totally from the first to the end, I think you will be totally cleared about the general overview and concept of viral replication. So I hope this class will help you. So let's start. So we know that viral replication cannot undergo by binary fission, because binary fission is in the case of bacteria. So in case of viruses, they go 7 sequential factors. What are they? Attachment, penetration, uncoating, biosynthesis, whether it is a DNA, RNA or protein, then assembly, maturation, and then after all release. This release will help in infecting the other cells as well as most of the cells of the body in case of humans or animals. Or in case of a in vitro culture, in vitro means, who don't know, in vitro means when you are working with a bacteria or a cell line or animal cells outside the body, that is in the laboratory condition, this is called in vitro conditions. So in vitro conditions as well as in vivo, that is opposite of in vitro, in both the cases I will give you proper definitions, explanations about the replication. So remember these 7 steps which will happen, which is bound to occur even if virus needs to success in their replication process or replicating their progenies. So these 7 sequential steps. Now the viral reproduction pattern, this graph, as you can see here, this graph, it looks like the growth curve of bacteria, but there are differences, huge difference between a viral and a bacterial growth curve. So if you are culturing a bacteria virus in vitro condition, you will be clearly observing this graph. So this is the pattern of their replication. So you can say that in the 7 steps, which is the step, this one. So this is the total overview of the replication process. So if a virus attacks a bacteria or an animal virus, what you will be observing in vitro conditions, how the graph will be there if you are working with the virus. So here you can see mostly 4 steps, that is eclipse period, latent period, rise period, and then the total burst size. So one by one I will clear your concept. So mostly I have given here example of E. coli and phage. You know bacteriophage is a virus which attacks bacteria. So this virus is only infecting bacterial cells, not animal cells. So suppose this is the example of phage count in the x-axis and y-axis gives you the time in minutes, right? So the eclipse period is the initial portion of the latent period which host cells do not contain any complete infective variants. So what is the exact meaning of this line? So the meaning of this line is when a virus enters into a bacterial or animal cell, that time it will shed its capsid, or it may enter with its capsid. Capsid means the outer compartment of the virus. If you have not seen my part 1, definitely check it out first, part 1, then you come to part 2. It will be easy for you to understand the flow of virology. Now, if it entered without any capsid, then what will happen? It has only the genome. So actually there are no count of complete virion. Virion means a virus. So when a virus enters into a bacteria or a animal cell, that time the single one, the parent virion, you cannot detect because it is not in its complete form. Whether it is enveloped virus, non-enveloped virus, a bacteriophage, whatever you say. So that is why it is the eclipse period is the initial time in a particular environment. It may differ from virus to virus, bacteria to bacteria, or animal cell to virus relations. Okay, so here you do not contain any complete, as it do not contain any complete virion. Complete means the genome with their outer capsid structure, it may, or envelope structure. Now coming to latent period. So latent period is the next step of eclipse period. So eclipse, as I have told you, no virion, complete virion, but the scenario changes in case of the latent period. Here you have complete, means the cell, either it is a bacterial or animal cell infected, complete virions are present, maybe 500 or 1000 or 5 lakh or 5 crores, whatever the situation is, but they have full phased and fully formed virions in case. Okay, now after this latent period where all the virions are present within the cell but none are released, that is very important. So here none are released. So remember, in the latent period, till latent period, you have virions which are not released in the medium, whether it is in vitro or in the cell, outer of the cells, so that it can infect the other cells, or in the medium, right? The latent period ends with the host cell lysis and rapid release of virions. That is called the rise period. So after the latent period, means just before the lysis, up to this is the latent, and after that it is the rise period. So rise means it started rupturing the cells, whether it is a bacteria or animal cell. I am repeating again and again because you can compare in your mind both bacteria and animal cell. Things are going almost similar, but there are differences. Definitely there are other differences considering the 7 steps. So in this rise period, cells lysed and rapid release. Now here 2 lines are given. So what is the meaning of these 2 lines? Blue and red. So blue line depicts that you have the virion which are not free in the medium, right? Total number of complete virions and free in the medium, both intracellular and free in the medium comprises this blue line. Okay, and here virions that were free in the culture and had not infected any other host cell, plus those virions that were released from infected cells. So all complete total count of the virions in that situation is depicting in the red line. So I hope you are clear with the blue and red line. So blue line, again I am telling, or you can see, pause the screen and read it. Total number of complete virions, both intracellular and free in the medium, both intracellular and free in the medium. But in case of red line, the number of free viruses, mostly free viruses, virions that were free in the culture and had not infected a host cell, plus those virions that were released from the infected cells. After the rise period, there is the size of this total count of viruses is called burst size. Okay, so I am first depicting in one slide this bacteriophage life cycle. So bacteriophage can be of 2 types, virulent and temperate, that you all know. So virulent phages infect their host bacteria and lyse them, that is virulent, and temperate means they will infect their host bacteria, but it can be present as an integrated form of their genome into the virus, into the bacterial genome, and carry on their progeny from one cycle to another cycle with the bacterial replication. and later in any stage or with some induction they can go for lysis. So lysis or the virulent phages or the virulent viruses or the virulent bacteriophages has only one option, infect and lyse. But in case of temperate, they have two options. They can either remain silent within the bacteria, integrated into their genome and replicate with the genome one by one or cycle by cycle, and then at some point of life stage with some induction or any kind of environmental trigger, they will go to the lysis stage. So that is temperate phage. Now that I am telling here, as you can see here, this bacteriophage DNA, if you consider this the first step, it's seated on the receptors present on the bacteria, giving its DNA. The capsid do not enters into the bacteria, only the genome is entering into the bacterial cytoplasm. So the bacterial chromosome you can see here. Okay, now in the lytic cycle what will happen, this genome will replicate itself, right, by hijacking the host mechanism using the host polymerases, either DNA polymerase, RNA polymerase or the protein machineries. Then they will form progeny bacteriophages, means other numbers of different numbers of other bacteriophages, means they are baby bacteriophages. They will lyse the cell and then again it will released into the medium or in the surrounding environment infect another bacterial cell. This is the case in case of lytic cycle. But what happens in case of lysogeny? Here you can see this genome. Again the first step will be same, attachment, then penetration, then the genome is there which will be undergoing replication. So in this stage you can see that the red part, red is the bacterial DNA and this blue is the bacterial genome or nucleoid. Now the red is integrated into the bacterial genome, and this step or this phenomenon is called prophage. Okay, so you can see here prophage is simply the viral genome either integrated into the bacterial chromosome or free in the cytoplasm. So in some cases, very rare bacteriophages has the capability to remain isolated genome in the cytoplasm and replicate itself for making progenies. Some bacteriophages have this capacity. For them, that stage is also called prophage. But in general, if I say, in general, in lysogenic cycle, most of the phage DNA integrated into the bacterial DNA, and this condition is called prophage. Okay, very important step in case of lysogeny. Now this prophage DNA is copied. When it will be copied? As the bacteria divides itself by binary fission. So whatever the number of bacterial genome, same will be the number of viral genome, because it is integrated. So it is combining the function. Now this prophage will, what will happen due up to at some point induction or exposure through stress such as UV lights, excision will form. Excision means the prophage will get out from that bacterial DNA and this will initiate its replication and progeny formation, and it will lyse ultimately. So it gives upper hand to the bacteriophage or the virus, because in some situations when they cannot survive, that time they can hide within the bacterial genome. In a later time when everything is suitable or the number of already increased number of their genome, so the number of variants will be higher also. So temperate phage has its own advantage. Now the triggers which will compel the lysogenic genome to excise out from the bacterial DNA is called the process phenomenon called induction. So the triggers, after the triggers or the trigger mechanisms or trigger channels like ultraviolet radiation or any environmental condition, this phenomenon is called induction. Okay. Now here also an important phenomenon takes place in case of lysogenic bacteria or lysogenic phages. Lysogenic conversion happens here. So what happens here? Alteration of the characteristic surface of epsilon phages. So epsilon is the example here. So consider this definition first. Involves alteration of characteristic surface receptors of the host or induction of pathogenic properties. So what is lysogenic conversion? I've given two examples here. First, it will change the receptor characteristics. How? They see the example epsilon phage, which is specific for Salmonella sp. So if one epsilon phage infect the Salmonella sp, Salmonella species, the first infection will change the receptor sites. So what will happen? The next epsilon phage will not be able to bind the same receptor, even if there is no epsilon phage present after the infection process. Let me tell you again, I think it became complex. So suppose one epsilon phage A infect a Salmonella. Okay. Now A epsilon phage completes its life cycle and then lysogenic life cycle, it will enter into the DNA of bacterial DNA, Salmonella DNA, integrated itself, and it is keep on dividing, right? In this situation, if again a new, suppose B epsilon phage comes and tries to infect that same Salmonella sp, where a prophage is present of epsilon phage A, it cannot happen because the receptors changed due to the properties of epsilon phage A. So A has make the necessary changes through protein expression into the bacterial cells so that the bacteria do not show the same receptor sites where epsilon phage A binds. That is why the next epsilon phages will not be able to bind. So this is a kind of lysogenic conversion. Okay. Now another type of conversion, what can happen? That it will give some property to the bacteria. What property? Induction of pathogenic properties like beta phage and Corynebacterium diphtheria. So in case of diphtheria, you have heard about diphtheria toxin. So that toxin can be only produced if the bacteria is infected with beta phage. The bacteria, Corynebacterium diphtheria, which is not infected with beta phage, will not be able to secrete the toxin. So that is the beauty of lysogenic conversion. So it is a very important phenomenon in case of lysogenic cycle. So you cannot see these processes in case of lytic or lytic cycle. Only in case of lysogeny, we will be able to observe this phenomenon. It's a very important, very useful phenomenon for our day-to-day life. Now coming to the 7 steps. As you have remembered the 7 steps, whatever you learned till now is the replication pattern. First slide I have told you replication pattern. Eclipse, latent, burst size. Okay, these are all pattern of their total replication process. But the 7 steps is mandatory for either bacterial viruses or animal viruses, because if one has to infect a bacteria or a virus or an animal cell, a bacteria or animal cell, it must attach first. It must grab the host cell first. Without it, infection cannot process. So the 7 steps is mandatory for all bacterial and animal viruses. So now what I am telling, or what I will be telling you, is you keep in your mind it is same for bacteria. The proteins are different, the receptors are different, but the meaning of these 7 steps will be same. Okay. So mostly I will be telling about animal viruses here or animal scenario. So animal cell scenario. So be with me. So plant viruses do not need attachment because they do not bind to receptors. They must rely on mechanical breaks or injuries to the plant body. Only through them they can enter into the plant cells. So remember, plants viruses do not need attachment. Now again coming to animal viruses. So attachment can happen how? With the help of receptors, right? So every cell in human, suppose I am telling everything about human viruses or human cells. So human cells have specific receptors. Receptors are nothing but glycoproteins or more of a different type of sphingoproteins. Several things are there. So I'm not going in detail about that. So these are bound by viral proteins or the protein present on the viral surface. Those are called ligands. So viral ligands can bind to cellular receptors. Okay. And the viruses have attachment sites on their envelopes or capsid proteins that bind to complementary receptor site present on the host cell surface. So this line tells you clearly that receptors are bound by the viral ligands, which can be any kind of capsid protein or envelope protein. So here I have given a representative picture also. It is a non-enveloped nucleocapsid virus which is attaching to a cell. Okay, this is the host cell. As you can see, the nucleus, Golgi, other apparatus, mitochondria is present. So this step is mandatory to initiate an infection or to start the progeny formation. Now coming to step 2 and 3 in combinedly. So I have given here combined penetration and uncoating because it is happening very quickly. So I have given this point in a single slide. So let's try to understand that attachment, then penetration, and this penetration step itself combines with uncoating. Okay. So the mechanism of penetration vary with the type of virus. As obvious, all viruses are not same. So their ligands will not be same and their receptors will not be same. Though their detailed mechanism is not yet discovered for each viruses, but some viruses are highly detailed information are available, and depending on that we can say the virus can penetrate into the animal cell or the human cell by 3 mechanisms. What are the 3 mechanisms? First one is phagocytosis, right? Second one is membrane fusion, and third is injection of nucleic acid. Means, as we have learned in the bacteriophage case, only nucleic acid gets entered into the bacterial cell. So here also some viruses are present. In that cases, no virus envelope or the capsid getting entered into the animal cell. Only the nucleic acid, the genome, whether it is a DNA or RNA, getting inside the cell. So coming to the first process, phagocytosis. It occurs through the receptors mediated endocytosis, resulting in the uptake of virus particles within the endosomes. So if you have learned in the cell biology the endocytic mechanism or endocytic pathway, that time you will remember, if you can remember that thing, that first a particle when it entering into the cell through phagocytosis, a vesicle formed around them, and that is called endosome. The pH is very low. So later part I will show you the phagocytosis endocytic mechanism for some virus. So there also it will be clear to you. So remember that endosome helps the virus in entering the cell, and the total process is phagocytic process. So endocytic pH or endosomal pH helps in some viruses to release its genome into the cell cytoplasm. So very essential process. So it depends on the virus characteristics. Now coming to the second possible penetration mechanism. So one thing more, in case of phagocytosis, both capsid can enter with, means nucleocapsid with the genome can enter. In case of membrane fusion, mostly happen with enveloped viruses, enter by fusion of the envelope protein with the plasma membrane. So in the plasma membrane they fuse, so they keep their envelope or attach their envelope into the plasma membrane host cell, so that only nucleocapsid enters into the cytoplasm. So the envelope remains outside the cell and the nucleocapsid enters into the cell. So later part, when it going to exit from the cell, it will again get its code or the envelope. So suppose you are entering in your room and you are keeping your coat beside the door. So again, when you are getting out of your room, you are getting your coat for the purpose you are going outside. So same thing happens in case of enveloped viruses or the membrane fusion technique penetration. In case of injection of nucleic acid, you can see here in case of bacteriophages, viruses that infect bacteria cannot penetrate the rigid bacterial cell wall. Only the nucleocapsid or the genome enters into the cell, right? Now, I think you are clear with the penetration mechanism, and during this penetration, uncoating happens, uncoating in the form of nucleocapsid or uncoating in the form of envelope. So penetration and uncoating are combined process or a follow-up process. Now here I have given some of the pictures. This will make your concept clear. So that's why I am giving this. So entry of enveloped virus by fusing with plasma membrane. So fusion. What happens in this case of fusion mechanism? You can see the spike proteins are present in the envelope. So first structure, the outer structure is envelope. Okay. Then there is a capsid structure. And the spikes are present in the envelope, remember. And after that, the third layer is the nucleic acids. First layer is the envelope. Second layer is capsid. Third layer is nucleic acid. And these spikes are nothing but the proteins present in the envelope. Now, the spike proteins will find its proper receptors in the animal cell. It will attach to the receptors. So this is an animal cell representation. Perfectly bound form. Now the envelope remains outside and the capsid with its genome entered into the cell. In this step you can see. Now this will start processing its progeny formation. By different steps I will be telling you in the next few slides. So entry of the enveloped virus by fusion. Okay. Now coming to the entry of enveloped virus by endocytosis or phagocytosis process. Here the same virus, envelope, capsid, everything, it will be entering into the animal cell. So total virion structure, nothing is left outside. Total virion structure is now inside a vesicle called endosome, which has a lower pH, and this total structure will be released into the cytoplasm. This endosome, in case of this endosome, the endosome enters the cytosol, but in case of some viruses, this envelope remains attached to this endosome. Why it attached to the endosome again? Here nothing is happening to the outer cell membrane. It is attaching to the endosomal cell membrane. Because when the virus will get out from the cell, it will again go through this endosomal process and then endocytic pathway, and total it will released from the cell. So this total fully formed virion will again enter into the endosome and will be released to the outside environment, and during that release this outer envelope spike proteins will again be bound to the nucleocapsid. Okay, I hope I am clear with the concept. Now coming to example of non-enveloped virus. So no envelope is present outside this virus, only capsid, then nucleic acid as it is DNA or RNA, and this is the receptor of animal cell. Now this capsid will bind to the receptors and this will totally enters into the cell. So nucleic acid is extruded from the endosome. Here also endosomal process is functioning, and the genome, only the DNA or RNA released into the cytoplasm. This is in case of non-enveloped viruses. Okay, so total structure is getting entered, but in the cytoplasm only the nucleic acid is given, right? And this nucleic acid will again form the total capsid structure and it will release from the cell. But here endosomal process may not take place. So as I am telling you, it is not possible for all viruses to accept the same mechanism. The outer mechanism is same, means the entry process is same, but the exit process or within the cell processes are different, or the receptors are different. So just try to understand the concept. Now coming to the replication mechanism, most important part of this study. Now coming to replication, we know the viruses can be of single-stranded DNA, double-stranded DNA, single-stranded RNA, double-stranded RNA. Now within the single-stranded RNA, it can be a positive sense RNA, it can be a negative sense RNA. So this much of virus replication pattern can differ. So one by one, slowly, I will cover each segment. The first variety of replication is single-stranded DNA and double-stranded DNA. So whatever I will be describing here will be implicated to either double-stranded DNA viruses or single-stranded DNA viruses. So before coming to that, I need to clear for you that I am sure that you know everyone know that the DNA to DNA is replication, DNA to RNA is transcription, and RNA to protein is translation, right? So in this total process, how the DNA virus is regulating itself. Now in this picture, I have given you a picture in this slide. You can see that it's a double-stranded DNA. You can consider it single-stranded also. Now first, what will happen? Early mRNAs will be produced. As it is DNA, you know that from DNA RNA is synthesized. So RNA polymerase will prepare or synthesize the RNA. Now the initial or early RNAs will form the early proteins. Now what these early proteins will do? It will hijack the host protein or RNA synthesis machinery for itself and start producing its own, means it will use some of the enzymes and proteins of the host and produce its own, means viral proteins and viral enzymes. Now these early proteins, it may be a RNA polymerase, it may be some other protein, it will help in replication of viral DNA. So if it is single-stranded, it will form single-stranded DNA. If it is double-stranded DNA virus, it will go for double-stranded DNA replication. Now within that several copies of the DNA or the replicated viral progeny, not progeny actually, it is a replicated viral DNA copy, some of them will be used for late mRNA production. Okay, suppose here it is this segment of double-stranded DNA is here being used for late mRNA and late proteins. So these proteins will help the virion or the progeny to assemble or manufacture the late proteins. Okay, and this process from RNA to again proteins is translation, right? I don't need to tell you again and again. So these late proteins help in assembly of the nucleocapsids. Within that, genome will be definitely present, either it is single-stranded DNA or double-stranded DNA. So this is the total process of single-stranded DNA and double-stranded DNA. Now few of the factors. I've given you an exception, that is in case of pox virus, they do not replicate in nucleus. Generally what happens in viral nucleic acid synthesis, it happens in the animal cell nucleus. Okay. But in case of pox virus, the genome is synthesized or the DNA is synthesized in the cytoplasm. So remember that viral DNA is synthesized in nucleus, but in case of pox virus, it is an exception. Now the transcription of, already told you, early mRNA and then late mRNA. Now this early structural that I have already told you, host metabolism, shutdown or hijacking, whatever you say. Now this progeny DNA will undergo several other viruses, it will incorporate within the nucleocapsid and several progenies will be made. So this is the common thing in case of single-stranded and double-stranded DNA genome synthesis mechanism. Now coming to the next. So within the RNA, there are single-stranded and double-stranded RNA. Now first, in the single-stranded RNA, there are positive strand or positive sense single stranded RNA virus or negative sense single stranded RNA virus. Okay, now that is separate issue what is positive negative, but still I am telling you that positive means here the copy of RNA from where directly the proteins can be synthesized. Okay, the proteins which are actually required to make more progenies or to divide or to replicate the RNA itself is called positive sense RNA. It is the real copy of the gene, which is, because we know that in case of DNA, suppose this is a double stranded DNA, ATCG is present, then the opposite what will happen? TAGC, right? So in case of positive sense RNA, suppose there are AUCG, which is required for specific protein synthesis. So, but if you present it with the complementary strand UAGC, it is called the negative strand, because UAGC cannot code the protein or the enzyme required for the particular function, either it is transcription, translation or replication. But the positive strand, which is the actual RNA copy or which contains the information to make the actual protein, either it is an enzyme or any other structural or non-structural proteins, is positive sense. I hope I am able to make you understand. So I have written here clearly also, in positive sense RNA, their RNA has the same polarity as mRNA. So you can consider it as mRNA. It will be easy to remember for you. So this positive strand RNA viruses are having a ready-made form of mRNA which can directly gets translated into proteins, and many other copies of more mRNAs or more positive sense RNAs. So what happens here? First you can see at first the positive sense single stranded RNA, some of the early proteins will be made. Now this early proteins, it may be a RNA polymerase which will help in transcription, means it will make RNA copy from the parent strand. So what will be the daughter strand? The daughter strand is the negative sense RNA. So why we need to prepare this negative sense RNA? Because when there are synthesis mechanisms in DNA and RNA, the complementary will be produced. Exact copies are not produced. So if there is ATCG, then TAGC will be produced. You cannot directly make ATCG to ATCG, right? So here the single stranded DNA RNA will be transcribed to negative sense single stranded RNA, which will be a copy from where the enzymes will make again the positive sense RNA, more and more positive sense single stranded RNA, which will be used to make late viral proteins. Here you can see late viral proteins through translation, and this late proteins will help in the assembly and release of the daughter virions. So I hope you understood that positive sense RNA immediately will follow some protein synthesis, and this protein synthesis, it is a RNA polymerase, viral RNA polymerase, which can be other proteins also. This will make more copies of negative single stranded RNA, and this negative single stranded RNA will individually be the copy from where original copies of positive single strand will be made, right? So this negative single stranded process or the step is mandatory. If it is negative sense, more positive sense will be prepared. If it is positive sense, more negative sense will be prepared, which will be the replicative forms. You can also say the replicative form is a term which will be copied directly to the actual genome. Okay. Now I have written for simplicity here positive sense RNA to negative sense RNA to again positive sense RNA, single stranded RNA. Now coming to the next part. So within the RNA, within the single stranded RNA, now coming to negative sense single stranded RNA virus. So as you are clear with the previous slide, that negative sense RNA means it cannot directly act as an mRNA. So what I have to do? I have to first make it a positive sense RNA. So first, this negative sense single stranded RNA with the help of viral RNA polymerase will be transcribed to positive single stranded RNA. Now this positive single stranded RNA from there the proteins will be made. You cannot directly make the proteins, means this will not, as it is not the direct copy of mRNA, you cannot directly make the proteins from here. So you have to make the single stranded positive sense RNA or the complementary strand from the negative sense RNA, and then from there the transcription will take place, translation will take place, and necessary proteins and enzymes will be formed, which will again help to form the more negative sense RNA, and that will be packaged with the proteins. So the final virion assembly will take place, right? So you can pause the slide, each slide, and try to understand properly. Okay, I have written in the simple words here also so that you can understand the fact. The positive strand, single stranded RNA translates to form proteins, and also it acts as a template and undergoes replication to form more copies of negative single stranded RNA, right? It's easy, just you have to remember the picture here, right? Now this is a little bit complex, that is double stranded RNA viruses. So double stranded RNA viruses have, most of the cases they are segmented, right? So till now what we have studied, none are segmented genome, either a single copy of single stranded RNA positive sense or a single copy of negative sense, or double stranded single copy or single stranded single copy of DNA, right? Now first we are coming to double stranded RNA viruses where their genome is segmented. Now segmented, what do you mean by segmented? See, the genomes of viruses in family Reoviridae, here I have given an example, Reoviridae, contains, Reoviridae is the family, within that Reovirus is present. Now 9 to 12 segments which are grouped into L, M and S. So for your ease of understanding, I have given a short picture here. See here, 3 of medium, 3 copies of large and 3 copies of single stranded, small. So both are double stranded RNA. So each segment will produce each protein, specific proteins, right? So the positive strand RNA act as an mRNA. So as you can see, I think you are clear. If you are clear with the previous slides, this is double strand RNA. So what will happen? First it will be separated into 2 single stranded RNAs. Then from there one is positive sense, one is negative sense, right? So the positive sense RNA or the positive sense single stranded RNA will directly act as an mRNA and synthesize the probable or the synthesize the actual proteins which is required for transcription and translation, or the early proteins or early enzymes, whatever you say. So same happens here. So you will get both positive sense, negative sense RNA copies and positive sense RNA copies. So positive sense will be used for translation as well as transcription, and negative sense RNA will be used for making more positive sense RNA as well as for packaging inside the nucleocapsid as a genome copy. So more positive, negative to positive, and then positive RNA sense to negative RNA sense. This both will again combine at the last and will be packaged to each virion particle, okay, with the nucleocapsid. So whatever you learned from the previous 2 slides, negative sense and positive sense, you just combine it here and try to understand. Okay, the main thing you need to understand is the segmented genome part. Okay, so the example here is the rotavirus I have given here, double stranded RNA virus, which is having at least 9 to 12 segmented parts of their genome, which these are not same parts. Okay, suppose if I mark them from A, 1A, 2A, 3A, then they are different. The polypeptide chains will be different, the enzymes will be different, the structural or non-structural proteins that enzymes will process different. So these are not copies of each other, these are totally different segments, okay, having different works and different genes, different nucleotide sequence. So that's very important. Now coming to retroviruses. So this is the last part of the replication varieties. Retroviruses, famous example HIV, you know that. So there are other viruses also. So HIV and other retroviruses have non-segmented single-stranded RNA copies. maybe 2, mostly it contains 2 single-stranded RNA copies, that is non-segmented. Okay, and the enzymes such as reverse transcriptase, they contain, why they are unique? Because they contain reverse transcriptase and integrase, two important enzymes inside them. It is not manufactured in the host cell, but it contains within the virus. Okay, they can prepare later part, they can again prepare it when hijacking or overtaking the host machinery, but the initial works or the most of the works are done by their own viral reverse transcriptase and integrases. So let's understand one by one. So they have a huge advantage of positive single-stranded RNA, right? Because it can directly act as an mRNA. So this positive strand RNA will be transcribed. So how that RNA-dependent DNA polymerase? Because it is a RNA genome, from RNA you have to make a DNA. Because it is a retrovirus, so from RNA they have to make a DNA. So which enzyme is required? RNA dependent DNA polymerase activity of RT enzyme. Here RT means reverse transcriptase enzyme. So in the next step, what you have? You have one copy of RNA, one copy of DNA. RNA is from the parent virus or the parental genome, and DNA is newly prepared, right? Now this RNA will be digested through ribonuclease. Ribonuclease is an enzyme which has a function, which contains the function of digesting RNA. So in this duplex, this RNA DNA hybrid or duplex, whatever you say, from there RNA will be digested with the help of ribonuclease enzyme, and single-stranded DNA will be left, right? Now from this single-stranded DNA polymerase activity of the reverse transcriptase enzyme. So try to understand, reverse transcriptase has two capacities. First, they can make RNA to DNA. Simple. Now later part, they can make DNA to DNA also, right? So the DNA polymerase activity of the RT enzyme, that is the single-stranded DNA which is newly prepared, will be made to double-stranded DNA with the help of DNA polymerase activity. How unique, huh? Now this double-stranded DNA will be transferred to host cell nucleus, as I have earlier said you, that the nuclear or the genome replication takes place in the nucleus of the host cell. So nucleus, there will be both the host genome or the host DNA and the viral newly prepared double-stranded DNA. Now this viral double-stranded DNA newly prepared will be integrated into the host chromosome, means the animal or the human cells DNA will be integrated, like you have studied in the prophage. Try to remember that. I told you many things will be similar, okay, but the mechanism is different. So in here, if you do not say this prophage activity, you can say it integrated genome. So the viral double-stranded DNA, which is made from a single-stranded copy, that is the initial parent copy, from there it make a double-stranded DNA copy, and that is now integrated into the human DNA or the genetic material, right? So whenever the cell divides, this machinery will be divided, or the viral DNA is dividing, right? Whenever the cell is replicating, or whenever any kind of enzymes is producing or proteins are producing, or when it is transcribing or translating, viral DNA will be transcribed and translated. Now this double-stranded DNA gets integrated into the host genome. I have already told you. From there, what will happen? Transcription will take place. Host RNA polymerase will now function, and it will not differentiate that this is the viral DNA or this is the human DNA. That host RNA polymerase will recognize it as a human DNA, and it will prepare more RNA copies from the DNA. Within that, human genes will be present, as well as the viral DNA. From there, viral genes will be transcribed to RNA also. Same occurs for the protein case, because from this mRNAs proteins will be produced. Now this total machinery will help to generate more viral single-stranded RNA copies, as well as more proteins that will be assembled into new variants, and then it will be released into the side by side cells. It will be more efficient infection. That is why drug against this kind of retroviruses is very, very difficult to make, because they totally submerge themselves into eukaryotic cells. So our machinery cannot differentiate or cannot degrade or cannot say it is the HIV DNA you should not replicate, you should not transcribe, or you should not translate. That is why it is a dangerous virus to acquire. Now, this is the total mechanism of retrovirus replication mechanism. Now, just remember these two enzymes, reverse transcriptase and integrase, which is very, very important for functioning of HIV or related retroviruses. Okay. Now coming to the assembly and maturation part, the sixth step, then the release is there. So here I have given two examples. This is the animal cell, right? This is the animal cell where you can see the capsid viruses, progeny capsid viruses, within that nucleocapsid genome is present. So this will be released. And here I have given example of bacteriophage assembly. So in a separate class I will show you bacteriophage structure. The bacteriophage having several structures. They have head, neck, tail, pin, different types of base plate, different types of structure that all need to be assembled perfectly to function as a functional bacteriophage, and then it will release from the cell. Okay, so this total part one by one will join. It's like the manufacturing will occur inside the cell, right? So whenever you buy a bike, different parts are made in the factory, but all are assembled to form it a functional bike or motorcycle or a car. Same thing happens in case of viruses. So their proteins are made separately, RNA is made separately inside the cell, and all are being assembled in some point, and then it is totally now functional to make itself a infective, particular, healthy variant for themselves, and infective for us or human cells. Now, here some example exceptions. DNA viruses are assembled in the nucleus. DNA viruses are assembled in the nucleus, except there are exceptions like hepatinoviruses and pox viruses, which assemble in cytoplasm. Same case for the DNA synthesis, already I told you, the assembly is same also. Now the maturation of daughter variants takes place either in the host cell cytoplasm, nucleus, or membranes of Golgi and endoplasmic reticulum, or the plasma membrane. Anywhere it can mature. Anywhere, some parts, suppose some parts are in the cytoplasm, some parts are incorporated into the cell membrane, some parts in Golgi apparatus, but from there every part will be assembled. Okay. So mostly plasma membrane contains some of their parts, because during release from the cell, the virus will contain it as an envelope. Okay. Or when it is releasing through the endocytic mechanism, it will get some membrane. Right. And the final step is the release. So the release of daughter variants occur by either lysis of the host cells. Okay, in case of bacteriophage, lytic bacteria, they do not have any option. They have to lyse the cell. So here I have given a picture of T4 release by lysis. So this is the bacterial structure. This is the transmission electron microscope picture. So this is why looking like that. So this is a bacteria, and inside that you can see these are small bacteriophages which are releasing already in releasing form or released form from the T4 from the bacteria, E. coli bacteria. So there are several proteins involved during release, right? So few specific proteins I will mention here, like lysozyme. We have studied for different cases. So lysozyme, you know that it attacks peptidoglycan. Now bacterial cells are made of peptidoglycan, right? So this enzyme will degrade the peptidoglycan, and it will degrade the host cell wall, and new progeny bacteria will the virus will be released into the surrounding environment. Another one is the choline. Choline is also an enzyme protein or enzymatic protein which creates a hole in the peptidoglycan, right? And it will help in releasing the variants. So two important enzymes you need to remember, lysozyme and choline. So if my classes are really helping you in your studies and it is really clearing your concept, and if you really like my classes, give a big thumbs up and don't forget to subscribe my channel BioAffairs. This type of subscription and likes give me the enthusiasm to prepare more futuristic classes for you guys, to make your classes more conceptual and clear your concept. This will help you in future in clearing your national level exams as well as job exams. So thank you very much. Be with me.